Hey there, GATE aspirants. I'm sure your preparation is going on in full-fledged for the February exam. Well, Biotechnica is back with another important topic from GATE syllabus. Today I'll be discussing protein conformations. Yes, protein conformation, it is from the chapter section 2 from biochemistry syllabus subject, and this is a very important concept because protein structure is required for many other units as well understanding, and even it is important from question point of view. So let's jump into the topic right away. Yes, we'll be discussing about the properties of protein conformations, why it is required. Then we have several levels of protein structure. Proteins are not directly folded into their functional form. There are some hierarchies, means level by level. The protein sequence polypeptide chain is folded into a functional protein which we called as the native protein. Now, thus these levels of folding are primary structure, secondary structure, tertiary structure, and quaternary structure. In between them, there are some other structures as well. For example, super-secondary structure or motifs, domains, and some other secondary type of structures which I'll be discussing with you. So briefly telling you about what are these levels. Primary structure is simply the polypeptide chain sequence which is in the linear form. So as you can see, this is almost in the linear form. It's just the sequence of the amino acids which are joined by peptide bonds. Now this is called as the backbone of the protein or the backbone of the polypeptide chain. Now when this backbone takes a certain kind of shape, as you can see here, this backbone has taken the form of a helix structure. Or here the backbone is in the form of a pleated sheet structure. So in that cases, those structures formed, they are called as secondary structure. That is the next level to second level of structure, right? Now several such secondary structures, they may come together. As you can see, you have some helical structures, you have some sheet structures, you have some turns or some motifs or some domains. The entire polypeptide chain, starting from its N terminal to its C terminal, the entire polypeptide chain, whatever overall 3D structure it assumes, overall means including all its components, including all its fold components, that is referred to as tertiary structure or the third level of folding. Now sometimes most of the proteins, they will be functional till here, means in their tertiary structure itself they will be fully functional, as this type of folding exposes the right types of functional groups on the surface where they can interact with some other molecules. That's what function is all about. The interaction with some hormones or some cofactors or some ligands, signals, or if it is an enzyme, the interaction with substrate. So that's what the function will be. It will be decided on the structure. Now, but there are some proteins which might level another level of folding, which is the quaternary structure or the fourth level of folding. For this, the several tertiary structures, they will come together and they will form a association. So quaternary structure is more of like an association. It's not that individual chains are simply entangled between each other to form quaternary. No, that's not what it is. For quaternary structure, it is more of a association between already folded subunits. This entire thing which is fully functional will be called as a unit, and the individual polypeptide chains which are already in their folded form, they will be called as subunits. Several subunits simply assemble together to form the quaternary level of structure. But however, not all proteins will have this quaternary structure. Some will be functional till tertiary itself. Now let's jump on to the other properties. What are protein conformations? Yes, they are linear sequence. What are the properties? When you have a polypeptide chain, that is a linear sequence of the amino acid residue in the polypeptide chain. Linear sequence means just the arrangement of amino acids one by one. as it is translated from the machinery. So after that, whatever structure it is taking, it determines the 3D conformation of the protein. Whatever is the linear sequence, that will decide in what form the protein is going to take the 3D structure, which will be the functional form. And that in turn will determine the function of the protein. So the structural conformation of a protein determines its function. How this function you will be getting? By how the protein is folding into the 3D form. And what will decide how the protein is folding in 3D form? It will be by the linear sequence of what type of amino acid is present. Because depending upon what type of amino acid is present, the secondary structures will be decided, or it will be taken. Depending on what secondary structure it is taking, the further super secondary or the motif or the domain structures will be taken, and finally that will decide how the overall tertiary structure is going to be. So as you can understand, the function is not directly related with the sequence, but yes, indirectly, because the structural conformation decides the function, and the structural formation, it depends on the linear sequence. Structure of protein is divided into 4 levels of organization. So let's see which are those 4 levels. Yes, primary structure. Let's talk about this first. So primary structure, it consists of the amino acid sequence along the polypeptide chain. As you can see, these are the amino acids which are joined by peptide bonds. As you would know that when you have 2 amino acids, so central carbon C alpha, from one amino acid you will have this carboxyl group. From another amino acid you will have this amino group. Now from this amino group, a water molecule will be coming out, or it will be lost. So that's how this particular bond which is formed between two C alphas, this is what is called as a peptide bond. Just because there is a loss of water molecule, this formation of peptide bond, it is called as condensation synthesis, or you can also say dehydration synthesis. It's a condensation reaction. D means removal, hydration means water. This type of reaction, it is an endergonic reaction which actually requires free energy, peptide bond formation. This happens through the translation machinery on the ribosomes, and the primary structure is just a chain of amino acid that actually makes up the protein. Okay. Now these amino acids, yes, as you can see, this is one amino acid residue. We have 20 standard amino acids. So this entire polypeptide chain will be consisting of this 20 amino acids. What does the backbone consist of? Yes, you start with a free N terminus. We always write the sequence from N to C terminal. You have to memorize that. Just how for nucleic acids we always write from 5' to 3'. Similarly, for proteins, polypeptide chain, we always write from N terminal to C terminal. What is the backbone of the polypeptide chain? You have a free N terminal, that is NH3+, then you have a C alpha, then you have the peptide bond. Then you have another C alpha, then again you have the peptide bond. So like this it continues until it meets the free carboxyl end. And from the C alphas you will have the R groups, different R groups which come out. So amino acids, they are joined by peptide bonds. Yes. So how does a peptide bond form? It forms between the COH group of one amino acid, as I mentioned, and the NH2 group of the next amino acid. So that is a type of condensation or dehydration synthesis. Linear sequence, which is always written from N terminal to C terminal. And the peptide backbone, it consists of C alpha atom and peptide bond. Each protein, they have their own set of amino acids. So they are assembled in particular order, and depending on the sequence, what type of amino acids are there and in what sequence they are present, how much composition they are present, that will decide what secondary structures this is going to form. Okay. Now moving on to the next part, which is secondary structures. Now secondary structures, these are distinct, repetitive, local structural conformation. As I mentioned, their local structural means what? Let's say your polypeptide chain started from here, okay, and let's say your polypeptide chain is continuing till here. Now you can see this part, which is a continuous part. From here to here, you can see that there is a particular shape which is assumed by the peptide backbone. The particular shape is there, in which they appear like kind of a twisted sheet. Some pleated sheet is present. This type is a characteristic. It's a characteristic and repetitive. It's not that only one type of sheet is present. So again, there is a repetition of that structure, right? And this is pretty distinct. You can recognize it that, okay, it is different from this type of structure. This is another type of structure. So all these are keywords. You have to remember the keywords for all the secondary, for all the type of conformations. These are distinct, characteristic, repetitive, local, structural conformations. I will add one more point of the backbone. Okay. So one point to remember for secondary structure is that R groups have nothing to do with the folding of secondary structure. R groups simply remain unassociated. They will just come out of the secondary structures. For example, in alpha helix, the R groups, they come out of the helix. Okay, they are protruded, they are projected outward. Similarly, in beta sheet also, they are above and below the plane. The R groups, they are above and below the plane. The bonding which will stabilize the secondary structures. So obviously, if I am folding the backbone, I need some special type of bondings. Okay, so what are the bondings present in secondary structure? They are stabilized by regularly repeating pattern of hydrogen bond formation. So remember, secondary structures are stabilized by mainly hydrogen bonds. Now, between what? Hydrogen bonds are between the backbone atoms. So what are the backbone? As we know, we have the peptide backbone, and in fact, if you have so this is a continuation of the chain, and then you have some other CONH bond. So what will be the hydrogen bond? As we know, for hydrogen bond, we need a donor which is bonding to a acceptor molecule. So here this NH group serves as a donor. This is linked with the CO group which acts as the acceptor. So that's how these forms a hydrogen bond. Okay. Similarly, this can be bonded with some other bonding. So there is a pattern, there's a specific pattern of hydrogen bond formation. For example, if you see this alpha helix, how does it look? This part is zoomed over here, and you can see there is a bonding between the NH of one peptide bond and the CO group of another peptide bond. So this is the peptide bond. The CO of one peptide bond joins with the NH of other peptide bond. And that there is a particular pattern. You can see these dots, these are hydrogen bonds. Similarly, if you take a beta sheet, in the beta sheet you will notice that this NH group is bonding with this CO. This CO is bonding with this NH group. So again, there is a particular pattern in the sheet. Like, how are these structures stabilized? From a linear structure, if they have to be stabilized, which means they need some bonds between them. Okay. Very important point is that the side chains are not involved in secondary structures. Okay. Now let's look at what are the types of secondary structures. So all this, yes, the more popular ones are alpha helix and beta sheet. But apart from that, you also have beta turn, reverse turn, random coil, and even loops are there. Let's look at them one by one. First, let's talk about alpha helix. So as you can already see on your screen that alpha helix has a helical pattern, like this the helical pattern goes, and mostly this is in the right-handed form. As you would be knowing that the type of amino acids that we have in proteins are L amino acids. And L amino acids, when their backbone is folding, there are some angles which are called as phi and chi values. These can take only some particular angles which promotes the formation of a right-handed alpha helix. So remember, the right-handed alpha helix is what is mostly you will see in proteins which are composed of L amino acids. The left-handed form is not preferred. Okay, only if there are some specific bonds, additional bonds to stabilize, then only left-handed will be preferred, else right-handed is what is most preferred. Then where are the side chains coming out? As I mentioned, the side chain of these components, they come out of the C alphas. So different C alphas are there. From there, the R groups are coming out. Now, depending on what type of R groups are present, the stability of these alpha helixes will be accordingly modified. Every helical turn has 3.6 residue. Now these are having some fixed dimensions. Okay, these are not random structures. These have fixed dimension. Especially alpha helix is having more fixed structure than beta sheet. Beta sheet is a little bit more breathable structure, but alpha helix is a more fixed kind of structure. So what is referred to as one turn? So this is one turn. Mathematically, what you can call it 360 degrees. 360-degree rotation is what is one turn. What is the length of this one turn? This length we call it as pitch of the helix. So pitch of the helix is 5.4 Armstrong. You have to remember the unit also. 5.4 Armstrong is the length of one turn. And how many residues are present in that one turn? There are total 3.6 residues. You have to remember that, okay? And what is the hydrogen bonding pattern? So if you take the CO of the ith residue can be any residue. It can be the first residue or the second residue or the 23rd residue. Depending on which residue you are starting, ith residue to NH group of I plus 4th residue. It means if your first residue hydrogen bonding is starting from 23rd residue, this we consider as I. So from here the CO group, whatever the CO group is there, it will interact with I plus 4. So I means it was 23. 23 plus 4 is 23, 24, 24, 25, 26, 27. So 27th group it will interact with, the NH group of 27th group. Okay. So this spacing has to be there between the hydrogen bonding. A minimum of 4 amino acids need to be there, amino acid residues, then only the hydrogen bonding will take place. So that was about the alpha helix. Now moving on, the beta sheet. So beta sheets, they are extended most linear segments called as beta strand. So they are, as you can see, this is a beta sheet. How it looks like, it is having some extended conformation, not the helical conformation what you see. So this is the extended conformation which you are seeing over here. Adjacent beta strands hydrogen bond to form a beta sheet. So these individual linear patterns, these are called as beta strands, and several beta strands they can join, means they can be stacked next to each other, where there will be hydrogen bonding between them, and this entire thing will be forming a beta sheet. Okay, so beta sheet can be of two types. This arrangement can be of two types. Sheet arrangement consists of pairs of strands lying side by side. So they can be just arranged next to each other, side by side, they can be arranged next to each other. Okay. R groups, where they will be present, they extend above and below the plane of the sheet. So as I mentioned, the R groups will either be protruded above or below the plane of the sheet. Right? So you can see this is the R group, this is the R group, these are R groups above and below. The two strands can be either parallel or anti-parallel. Okay, so it's about what directions the strands are going. Now what is parallel beta sheet? Strand directions N2 C terminus are same. Anti-parallel is strand directions N2 C terminal, they are opposite. So why, what is N2 C terminal? If one strand is there, it goes from N terminal to C terminal. Now then it takes a 180-degree turn. Okay, it takes a 180-degree turn, and then again the strand continues here, but the direction is now reversed. So this side is N terminal, this side is C terminal. Then there will be hydrogen bonding between them. This is what is called as anti-parallel arrangement, where the senses are not matching, means the N terminal and C terminal. Note that the hydrogen bonding pattern is linear in case of the anti-parallel strands. And another possibility is parallel beta sheet. So for that, if one strand goes like this, N2 C terminal, then that strand takes a 360-degree turn. Here it was taking a 180-degree turn. This was called as a turn. Since it is present between two beta strands, it's called as a beta turn. Okay, but this will take a 360-degree, and again it will go in the same direction. So this will be N and this will be C. Where these are matching, this arrangement is called as parallel beta sheet, which requires a crossover connection. Here the entire polypeptide chain is taking a 360-degree turn. So this is a crossover connection. And here the hydrogen bonding is at an angle. It's not linear like this. So the hydrogen binding pattern is at an angle. Now if you know about hydrogen bonding, if they are linear, then they are more stronger, they are more stable. That is why out of antiparallel and parallel, antiparallel having linear hydrogen bonds, they are more stronger and more stable structures. So that's what it is written. Antiparallel beta sheet is more stable due to the more well aligned linear hydrogen bonds. Okay. Let's come to the next type of secondary structure. It is beta turn and reverse turn. It is a segment, any segment of polypeptide chain which takes a 180-degree direction turn. As I gave example of beta turn, it is beta sheet. It is taking a turn and then coming back. So these are the sheets in between that you have a turn. Typically, a turn will be formed by adjacent 4 amino acid residue. So if you have one residue here, another residue here, another residue, another residue. So here there will be bonding between the first and the fourth residue. So this is first, this is second, this is third, this is fourth. So instead of writing first, because this is not the first amino acid of the peptide actually, this is some amino acid in somewhere in the center. So we will write this as ith amino acid, and the fourth one we will write as I+3 amino acid. Okay. So hydrogen bond in turn is formed between the ith amino acid and I+3rd residue. Next one is random coil. Next type of secondary structure is random coil. As the name is suggesting, there is no regular secondary structure. So what does it mean? That it can be any length. It can be around 4 to 20 residue long. As you had seen for alpha helix, beta sheet, beta turn, there was a particular pattern of folding. But in random coil, there is no real pattern. It is not characterized by any regular hydrogen bonding pattern. No regular pattern of structure, no regular pattern of hydrogen bonding. And this is mostly found at terminal ends of polypeptide chain. When you have a polypeptide chain, it's not that from the starting itself an alpha helix can form, then immediately a beta sheet. No. In the entire polypeptide chain, 50% of structures may be present in regular repetitive pattern, that is alpha helix and beta sheet. Rest all 50% will be in the form of turn, random coil or loops, because they simply cannot take those standard alpha helix and beta sheet. I mentioned that the type of amino acids determine what type of secondary structure it is going to take. So these are mainly found at the terminal ends or the parts of the protein which are not able to take any other secondary structures. Next are loops. So loops are not longer than 12 residues. These are unstructured regions found between the regular secondary structural elements. Now these are unstructured. Unstructured means they don't have a particular defined structure. Okay, they keep on changing. They are dynamic just because they cannot take a fixed dimension of phi and psi. So that's why these are present as unstructured areas. Let's move on to the next type of structure, which are called as super secondary structure or motifs. So what are these? As the name itself suggests, they are arrangement of two or more secondary structures for stability. For example, you can have a alpha helix and then a beta sheet. Okay, then again an alpha helix, again a beta sheet. Alpha helix, beta sheet. So this is forming a regular pattern, and two or more secondary structures, they are remaining together. Why they will remain? For stability. Okay, like that there are different types of, they are also called as motifs. Motifs are strictly for structural stability. Okay, there will be only few motifs which have a functional significance, like zinc finger motifs, leucine zipper motifs. But apart from that, most of the motifs, they only have structural significance. For example, this is also one motif, helix, turn, helix. There is a helix and then there is a turn and then is another helix. So this together it forms a motif. Or else you can have a helix loop helix. So you will have a helix like this, then a loop will be formed, a random loop, then again a helix. Okay, so protein 3D structure is completely a variable structure. There are no set rules actually. Yes, the overall structure, if you have a particular sequence, the 3D structure will always be the same, because the most stable structure will be its native structure. And every time a protein folds, it will come to that stable structure. But there are no general rules to fold. It completely depends upon the sequence. You can have alpha alpha corners, beta beta corners, and even beta hairpin beta. Next are domains. So what are domains? These are one level above the motifs, but I will not say several motifs form domains, not necessarily. A stretch of polypeptide chain can form a domain. So basically it will have a distinct functional or structural unit in a protein. And most importantly, these are independently folded, means they don't require the folding, they don't depend on other parts of the protein to fold. For example, if you have a stretch of polypeptide chain, maybe this part folds independently into some kind of domain. Then the rest part of the polypeptide chain folds independently. If you somewhat cut off this part, then this part won't be affected. This will still remain, it will retain its 3D folding. Even this will also retain its 3D folding. Okay. So this folding doesn't depend on this folding. These can serve as discrete functional units. Discrete means independently they can function. Okay. For example, you can take up a transmembrane protein. Now we know they will have some folding or domains outside, which is called as extracellular domain. The part which will bind with some ligands or receptors, growth factors. Then you will have a transmembrane domain, which passes through the membrane. Then you will have a cytoplasmic domain. So that will interact with some cytoskeletal components like actin, filaments, microfilaments will be there. So these three, even if you cut this off, that doesn't mean that transmembrane protein or cytoplasmic domain, they will get affected. Their folding will remain the same. Okay. But this entire thing is one polypeptide chain. This entire thing is one polypeptide chain. So domain can be a subcomponent of the tertiary fold. Okay. So it will be smaller than a subunit, but it can be independently folded. But not always necessary. Sometimes the domain itself is the tertiary structure. So the entire protein can be folded into just one domain. But then there are no other domains in the protein. So the domain itself is the 3D structure of the protein. But on the other hand, a protein may have more than one domains. And each domain will have a functional significance. That's the difference between motif and domain. Motif has structural significance, domain have functional significance. Moving on to the next category, that is tertiary structure. So what is tertiary structure all about? It is the overall 3D conformation of entire protein. Now you can see there are some helices, sheets, turns, loops, coils. So together they have taken the overall 3D structure. How they have taken it? There are some interactions between the side chains. So this is the polypeptide backbone, and the polypeptide backbone, from here some functional groups are protruding out, and those functional groups have some interactions with each other, thus leading to the stability of the overall 3D structure. So how are they stabilized? These are stabilized primarily due to interactions between R groups. As I mentioned, secondary structure is only by interaction between the backbone structure, but here the interactions are between the R groups. And these are stabilized by multiple non-covalent interactions. Several such non-covalent interactions stabilize these kind of structures. They will adopt the native conformation, which is the fully functional form. This form will be fully functional because it will expose the right types of groups on the surface. And this is determined by a variety of chemical interactions. So what interactions are there? One type is hydrophobic interactions. As you might be knowing, hydrophobic interactions, they are between non-polar residues which are present in the interior of globular protein. This what we call as a globular protein, okay, because it looks like a globe shape, a spherical shape. Here the amino acid residues which cannot interact with water, they will be buried towards the core. The ones which can interact with water or which are polar molecules, they will be towards the surface. Okay. And hydrophilic residues on surface, they interact with surrounding water molecules. So when you have non-polar groups, they tend to cluster together through hydrophobic interactions. This is the major driving force for the folding of the protein. This is the major type of bond stabilizing the tertiary structure. Another type of bond which you will see is disulfide linkages. These are covalent bonds between sulfur atoms of cysteine monomers. Now cysteine monomers, they need not be next to each other. they can be adjacent to each other. When the protein folds, as you can see, this is folded like this. But what is holding this protein in this particular shape? This is a cysteine residue from here and a cysteine residue from here, which together are involved in disulfide bonds. Another one is ionic bonds, which are found between some positive charge side chain and some negative charge side chain. Additionally, they can also have a more stronger bond, which are called as salt bridges. But however, salt bridges are not present in all type of protein. They are only seen in some extremophiles, where additional stability of the protein is required. Apart from this, you have hydrogen bonds, which are stabilizing the tertiary structure. Similar to that, what you see in secondary structure, hydrogen bond form is same, but it's just that in secondary structure you have exclusively hydrogen bond, but in tertiary structure hydrogen bond is just another one type of bond stabilizing the structure along with all these other bonds. Okay, so out of all this, these are all non-covalent forces. That means these are weak bonds, but together they give an overall strength except disulfide linkage. Disulfide linkage is a covalent bond. Rest everything is non-covalent. So they are comparatively weak bonds, but since there are so many bonds in the tertiary structure, overall they will be having a stable structure. Okay. And the last part is quaternary structure. So quaternary structure, it's nothing but the spatial arrangement of subunits in a protein. As you can see, this is a dimer. Now what is a dimer? Di means two, mer means molecule. Two tertiary structures, they have come together. This is one tertiary structure, this is another one. You can clearly see that they are complete tertiary structures, means independently they have beta sheet, alpha helix. So this is a complete subunit and this is another complete subunit. Between them there are some interactions. So between the subunit functional groups of one and another functional group of another subunit, there will be interactions because of which they stay together. And there is a particular arrangement in space, not that randomly they just accumulate or aggregate together. No, they have a particular spatial arrangement. These consist of more than one polypeptide chain, folded subunits assembled to form a functional protein. So this will give rise to a native protein which are stabilized by interactions with same as tertiary structures. So whatever we saw, hydrogen bonds, hydrophobic interaction, disulfide linkage, ionic bonds, same type of interactions will stabilize them. You may have homopolymer which are consisting of same type of subunits, or you can have heteropolymer which are consisting of different subunits. For example, you will have hemoglobin. Hemoglobin is a tetramer which consists of 4 subunits to be functional, which are alpha 1, beta 1 and alpha 2 and beta 2. So that's what they have, 4 different subunits arranged together. That's all for the important topic from GATE. As you can see, we have discussed several points and all of these points are equally important. Do know them and you will be able to answer the questions which are coming from this topic in GATE exam. All the best everyone. Thank you.